Our speaker this morning is Ronit Yarden from Georgetown. She is a cancer molecular biologist and is going to be talking about genetic epidemiology of breast and ovarian cancer. Ronit got her bachelor's and master's degree at Tel Aviv University, came here to Georgetown for a PhD, and then did a postdoc over at the NIH at National Human Genome Research Institute. Went back to Israel for a while, and then returned here to Georgetown, where she's been since 2007, I think. 2009. 2009 at the Lombardi Cancer Center. So, welcome, Ronit.
So, thank you for the introduction, and thank you for having me here.
You can pin it on.
Can you hear me?
Yeah.
Okay. So thank you for having me. It's a pleasure to be here, and it's sort of like a lovely and very collaborative community. So I will look into it with my kids. As Mark said, I was born here in New York. Then my parents, my mom is Israeli. She was born in Israel, pre-Israel, in Palestine days. My grandfather moved from Russia, from Ukraine to Israel in the 1920s. But my father is, his family is from Hungary, and he moved here, and they kind of met in between. And I was born here, then they moved back to Israel. I grew up in Israel. I went to my bachelor's and master's in Israel, and then I came here for a PhD. I wanted to try out how is it going to be back here. And then I stayed here for about 11 or 12 years, moved back to Israel, then missed Washington and came back, and I'm here. I have a connection with Georgetown because I did my PhD at Georgetown, and then I came back for a position at Georgetown. But I'm trying to keep up with my ties in Israel, so I'm trying to find ways to work them both together besides just scientific collaboration. So in the past year, in the last summer, I brought a class from Georgetown. We did 6 weeks in Israel. And Georgetown students, pre-med, came to do a fellowship in Israel in the Sheba Medical School Center, where I worked before. And it was a lovely program. There are blogs over at my website, so you can look at it if you want to see. And we're looking to do it again this year. So we try to make a connection between Georgetown and Israel, which would be nice, because it doesn't have a great reputation of being pro-Jewish so much. So, okay. So when I finished my PhD, when I did my master, I kind of went to do research about breast cancer, and I tackle it from the immunology kind of point of view, is like trying to raise the immune system to fight breast cancer. And then I moved to Georgetown for my PhD, and I looked at hormonal regulation of breast cancer. And so I continued, I've been working on breast cancer for many years, and then when I finished my PhD, they just found the BRCA1 gene. So I was all excited about it, and I moved to the Genome Institute to work on the BRCA1 gene, and since then I've been working on it. So a lot has been done, and there's still more to do. So what I'm going to tell you today, I'm just going to start with a very simple background, because I didn't know how well you are familiar with genetics. So just started about the genome. We all have multiple cells in our body. All cells have nuclei, and in the nuclei we have our DNA. Our DNA is built from chromosomes. We have 2 sets of 22 chromosomes. We call them sometimes autosomes, and they are. And then we have the sex chromosomes. For male, it's X and Y, and for female, it's XX. And the DNA is what we call the double helix. It has its building blocks are the nucleotides. And so we have 3 billion nucleotides in our genome, in each of our cells. So it's a lot of nucleotides, and it's, even though it's so many nucleotides, we only have 20,000 genes, which might sound a lot, but you know, like rice or tomatoes have about 100,000 genes. So not to say that they're more complex than human, but it's just to give you a point of view. So we have 20,000 genes in our genome, and they code for different proteins that. So if you stretch the DNA in the nucleus of every cell, it will stretch up to 6 feet of DNA, and it's if you would have to print it on a paper, it will be all the way up to the amount of paper will rise to up to the Washington Monument, and if someone would have to type it, it will take them 10 years if they work 7 days a week, 24/7. So it's a lot of genetic information there. So when our cells replicate and when we are transmitting our genes from our next generations to our kids, the DNA has to be copied and then has to be divided between old cells and new cells, and it has to be copied with high fidelity. But, so we have a lot of mechanisms in our cells to keep the DNA integrity intact and maintain that almost no mutations were introduced. And however, sometimes there are mutations, and mutations can cause different diseases. And so we all kind of have some variations in our genome, and we are all kind of mutant to some genes, but not all mutations are really transferred to causing a disease or having a physiological function on our body. But the reason why it's important also to have variation is that we are not all clones of the same DNA, right? So the introduction of some mutations or some variations over the course of the years in the population is important. So we're all a little bit different. If you look around us, we all look different, but we're 99.99% similar. And so that means that there's about 0.001 difference, which will translate, if you look at the 300 billion nucleotide, will translate about to 80 million nucleotides that are different between within the population, variations of. So it's not that we all have the same 80 million nucleotides different between each individual, but in the population overall, there could be at least 80 million variations in the DNA. So we have the building block for the DNA, and there are 4 nucleotides. I'm sure you, GATC, and they state for chemicals that are composed for a nitrogen based and a sugar and a phosphate. And so this is a sequence of a DNA. This is a partial sequence of a DNA, and you can say that there's a sequence here, GATC, that in different combinations. And some people would have an A instead of a G, or a T instead of a C, and so forth. So those variations, sometimes they are real mutations that have pathological consequences, but sometimes, as I said, they're just variation between individuals. Those variations will account for, why somebody has a brown hair versus a blonde hair versus a blue eyes versus a green eyes. These are not any pathological condition, but they're just variations in the population. So those variations in the population, we actually can utilize them when we study the genome, because they're like roadmaps. They're like road signs that tell us where are we within those 30 billion nucleotides. They give us, and we can locate them easily and use them as a guidance to find genes or to find association between genes and disease and so forth. So as I said, the variations, some of them are mutations, and we usually, the definition of a pathological mutation is that usually they are less than 1% frequency in the population. And those sites that are variations and are polymorphic within the population, we usually tend to, their frequency is more than 1%. And if they're somewhere between 1 to 5%, they are low frequency variation, and then beyond 5%, they are highly frequent polymorphic site. So, as I mentioned, there are two, each one of us get two sets of kind of genomes, one from our father, one from our mother. And they're not identical because of the variation in the population. And so, sometimes those variation in the populations, when they cause mutations, we can say that some of them are more impactful than others. And we can maybe say that if the mutation is dominant when it's one of the copies is defective and it's sufficient to cause a defective phenotype or a defective physiology. And we say that a gene is recessive or a variation is recessive when you need the two copies to be defective, not just from one parent, but from two copies to be defective. Then we call those traits recessive traits. And our genotype is the sum of our genes or the sum of our genome, and our phenotypes is what we express. So most of the time there's a correlation between our genotype and phenotypes, but as I said, some variations are not necessarily impacting the physiology or the phenotype. So they are just there, and we don't know exactly what they there for, or sometimes we do, but they don't have an impact. So if you look at, can you all see that? Or if you look at, this is a pedigree, this is how we in genetics and draw a family history, and the circles are females and the squares are males. And if they are filled with color, there usually means that this is an affected person or has the condition that we're looking for. So this will be a typical pedigree of for a family that has a trait that is a dominant trait. So, for example, Huntington chorea is a disease that was a phenotype that is dominant, or even the gene that I'm going to talk later on BRCA1 has a dominant effect. So basically, in this kind of families, you have just a mutation in one set of the genome, just in one version of it, and then it's sufficient to cause the disease. And you can see that the disease is passing from generation to generation. And it's about 50% female, 50% male, so it has no gender bias or anything. And then we also have mutations or family. Sometimes we have families that have mutations that are due to recessive genes or recessive traits. That means that you have to have the two copies of the gene to be defective. So you inherit one defective copy from your parental side and one defective copy from your maternal side. And since you don't have any functional copy, you would have a condition. So a lot of times these are a lot more rare situations to have two copies of the gene defective, and they come sometimes to life and being more frequent when you have marriage within families or consanguinity. And so when first degree cousins marry, you have more chances of chances for these conditions to be apparent. And that is true for small communities who tend to marry within the community, not necessarily first cousins, but if you have a small population that tend to marry within that community, that also will increase the chances of having a recessive disease. We know today about 7,000 diseases that are due to a single gene, like what I showed you here, and we know the cause of those diseases for about half of them. So there's a lot more to discover and a lot of more conditions that the genetic background is still unknown. So is Jewish a disease? Being Jewish is a disease. You know, we hear about those frequent mutation in the Jewish population, and especially in the Ashkenazi Jewish population, is it a disease? So it actually is not. There's no right. There's most of the genes that were discovered or were found in high frequency in the Jewish population and Ashkenazi Jewish population in the beginning are also later found out in other populations. So, and sometimes with a similar frequencies and sometimes with a little bit less frequency. But most of the Jewish people do not carry any more genetic disease genes than other population. So why is there was a high frequency of diseases in our variation in Jewish Ashkenazi Jews that was coming about in the beginning is because it was an isolated population. It was actually being Jewish, it's not just a religion, but it was a culture and it was geographically isolated. Whether it was in Europe and they lived in separate communities than the general population for many, many years, or was it in the Middle East, that they also lived in separate communities. So they tend to be culturally homogeneously settled and then not mixed with the general population for most part. So they were useful for genetic studies at the beginning because they were an isolated culture and they were somewhat homogeneous. But there are other communities or other populations that are genetic isolates, and the French Canadian, for example, is one of them. Newfoundlanders, people in Iceland, people in Finland, people in Sardinians, and in Israel also we have the Bedouin community, and these are nomads, people who live in the desert. And actually, when you go to Israel, and when I took the students this year for this genetic class that we did in Israel, we met with the Bedouin community, and they are even, and especially in Muslim and also other Muslim villages, they tend to be very to marry within their village. A lot of first cousin marriage. They have a lot of very, very specific disease to a specific village. And so when we visit Israel, we went to a Bedouin community that they moved from Egypt to Israel in the beginning of the 19th century. And because they were newcomers, they were not accepted by the other Bedouin communities, so they had to marry within their own. They had one woman that was deaf, and then that mutation propagated, and then they had 50% of their community are now hearing impaired. Because they were not assimilated within the other Bedouins communities or in Israel at the beginning, they kind of developed their own sign language. And later on, when Israel was established and they kind of got settled a little bit more, and they have now permanent buildings, they got into the general education system. So the new kids, they are learning the sign language from the regular sign language in Hebrew. And it's interesting because the old generation using the older sign language, the new generation is using the the Israeli sign language, and they cannot communicate between them because they don't. It's some different sign language. It was very interesting. They are actually. They started a program also to do genetic testing for themselves, and so to try to find a solution to dilute the problem of the hearing impaired, because they're still marrying within the community. And there are actually different approaches in Israel from clinicians on those consanguinity marriages. Some geneticists, some clinical some clinical doctors, geneticists accept it and try to work with the community to, they embrace that there's some importance for the village social network to keep that tradition. And so they try to work and introduce methods how to monitor and do genetic testing and but work around it. And some doctors are really against it, and they kind of try to preach them to avoid any kind of marriage that are in first cousins. So it depends on the doctor that is in the community around there. It's interesting. It's very interesting to learn about it. So anyway, so it's not just Jewish population that is isolated community. There are others there are other diseases that are in other communities. So those genetic variations I told you about that are in our genome, and they're very frequent, we can use them to track down the genetic, try to track down their locations and tell us about evolution and how we can, from there, decipher how changes in the genome happen over the years and how they relate to different diseases. And we can look at different populations and see migration and see admixture between different populations. If they have marriage between different populations, we can see how the contribution is, we can find out the contribution of different populations. We can track somatic changes, which are more what we have when in different cancer conditions, when we have mutations that happen not in our germ lines, they're not inherited, but they are happening due to exposure to chemicals or environment or our own metabolism. And then we can identify, as I said, they can help us identify genetic diseases because they might associated with or found nearby genes that are actually causing disease. And we can also now use them in a whole new field of pharmacogenetics as to use them as markers for prediction for response to different drugs, and then tailor different treatments to different patients in what we call precision medicine and the moonshot initiative, and which is a big thing now. Okay. So population genetics is the study for studying the genetic variation within the population and how it changes over time. And it examines the frequency and the distribution of those mutations in the different populations and the different. So each copy of a gene, we call them allele, and they have 2 alleles for each gene, one, as I said, one from the mom and one from the dad. So how variations introduced or into our population or to our gene pool. What we call is the gene pool is the sum of the genetic information that is within a population. And one way to introduce new variations are mutations, and those are, as I said, are causing some conditional pathological conditions, but sometimes there are just good changes like selection. If you have I live in societies that have lactose tolerance, in societies that have more adjusted to work with use dairy products for long, like in Scandinavia or Northern Europe, so they are more lactose tolerant than other populations around the world. Sometimes with migration, you introduce, if there is a variation that is moved, if it was a very close society, and then there's a movement of newcomers to the population, or if there's Icelandic population, it was isolated, but people move in and they introduce new genes into the population there. There will be introduction of new variations and new information. Genetic drift is what we call by chance. Suddenly some genes are, or some variations are lost. If there was some kind of a disease that some genetic variations were more susceptible, if it was, so it's basically by chance, and then there's founder effect, which I told you about, which is basically if there's a small community, there's one mutation, like in the Bedouin community, there was one lady that was deaf, and then it got amplified because they keep marrying it within the population. So founder mutations are those that happen in small communities that have, that tend to marry within inside a community. Looking at the, yes?
Can you explain the illustration at the bottom, the bottleneck?
Right, so the bottleneck. So if you have a population, for example, that have certain, this is the gene pool, the different beads, and then suddenly they're settled in some location and maybe they're moving to another location, but only a fraction of the population is moving, then some of the genes that might not be transferred together with the part of the population because they're not so frequent, then you have a new population that is just missing some of the original genes or the original version. So, and sometimes it's, as I said, it could be because migration of a specific fraction of the population, or it could be because a specific disease that affects, I mean, this is not just for human, but if a specific disease affects a specific type of certain genetic phenotype, certain gene pool, so that will affect, let me say it again. So if by chance you get rid of some of the genes in the population because of disease or because they migrated in or out, then you are affecting the what's left over or what migrated. Yes?
Yeah, the example you gave of the deaf gene being propagated, Oliver Sacks wrote a book called The Island of the Colorblind, and maybe some of you have read that, where there was some disease that wiped out most of the people on this island, and one of the people out there had color blindness.
One of the survivors.
What?
One of the survivors.
Yeah, so one of the survivors had color blindness, and now, and because there weren't that many people having babies after that, the island then ended up with a large population of people who have color blindness, just like the deaf example, and that's where the bottleneck comes in, where you don't have the broad population, so you propagate a particular gene.
So you enrich for a particular.
Ronit mentioned that this is not only for humans, so in animal populations, when the population gets too low because of hunting or landscape, like, I don't know if you know the example of cheetahs in Africa, but there's a big problem now because the cheetah population is so small that there's a lot of genetic homogeneity. There's really not very many differences between cheetahs because all of the differences have been wiped out with loss of the population. So that's the kind of thing that we're talking about.
Isn't there something similar to bananas?
And it could be in every organism. So
Yeah, that's because it's only one strain of banana that has been propagated commercially, so that all bananas essentially all bananas now come from a single source, and that single strain. And that strain is susceptible to a particular disease, so there's a
Right.
problem of potentially wiping out the large fraction of the commercial banana.
Right. It's for a genetic modified disease. That's what happens. Like you have a genetic modified disease, and it's being marketed for a large population and farmers, and it's being and then if there's a disease that happens, all the crops are being affected, and then it will affect the genetic combination of.
Could I just add one thing to that banana comment? So that strain of banana is actually infertile, so they can't propagate. They actually do cuttings to get new banana trees. So that's another way to bottleneck, is you have basically created an entire population of bananas that are absolutely identical because you were just cutting parts just like you would do at home. You know, so you've got a bottleneck where you basically lock in the genetic code there. We've already lost one.
Save the bananas.
So just a little bit about the Jewish history. So in the second century, or in the first century, second century, when they were moved out of Israel, they moved along the Mediterranean through the northern Africa and through the sea to Italy and then Turkey. And so there were Jewish migration all over around the Mediterranean, and building Jewish community. And then after the Inquisition in 1492 actually, there's the expulsion of Jews from Spain, and they moved back to the north of Africa, and then moved to Middle Europe, and then upper to the Rhine kind of valley, and where what we call is now the Ashkenazi Jews. And then from there they moved a little bit up to they went through pogroms and all kind of massacres. They were moving also towards Eastern Europe, into Russia and Lithuania and Ukraine. And so the Ashkenazi Jews are those Jews who settled around middle part of Europe, and then moved, some of them moved a little bit out to Eastern Europe. So when we do genetic studies on Ashkenazi Jews, these are usually the communities that we look into. And the Sephardic Jews are those who were expelled from Spain and moved into either the southern Mediterranean countries like Italy and Greece and Turkey, Italy, Greece. And then we have what we call the Mizrahi, or also Sephardic Jews who live on the northern part of Africa. So it turned out that even though Jews were spread over all over the around the Mediterranean into Europe, we're still very similar to one another, more than that we're similar to other populations. And when you look into the genetic composition of Jews, it's actually kind of a combination of Semite community or Middle Eastern community and European community and European genetic genome. So it's a combination of both these lineages, but we're still Jews, even Jews from Iraq or Iran, or Jews from Spain, or Jews from Ashkenaz, are still more similar to each other than to the other types of populations. So when they started doing genetic studies on Jews, and then they do the use the Ashkenazi DNA, they found there are several mutations that are several conditions and several diseases that are frequent in Ashkenazi population, like Thai-Sachs, which is about 3 to 4%, and then Gaucher is one of the most common one, is 4 to 6%, and some other metabolic diseases like Niemann-Pick or Canavan. But there are also what I'm going to talk later about is the BRCA1 and BRCA2, which are the breast and ovarian cancer genes, and also count for 2.5% within the Ashkenazi Jewish population. But they're not just in Ashkenazi Jewish population. They're right now for the Ashkenazi Jews. They're like a panel if you want to do screening, and if a couple wants to get married and they want to do a prenatal screening to avoid some of the genetic diseases. There is a panel in Israel that is doing for 38 different Ashkenazi diseases. And I think in Israel is one of the populations that test that test heavily for genetic conditions. It's covered by insurance. We have a national insurance, health insurance, and it's covered by the state, those genetic tests. So everybody's doing them, and everybody's doing almost the whole panel. So prenatal screening, as I said, is very common in Israel. It helps to screen for diseases and autosomal and autosomal diseases, but also for the less common ones, the recessive diseases, and avoid marriage, or not avoid marriage, but maybe plan your pregnancies in a way that you can maybe do a pre-genetic, so do testing on and do IVF and do testing on the embryos to avoid implantation of with genetic embryos with genetic diseases. Thai-Sachs birth went by screening went down for almost 90% than what it used to be, and in the ultra-Orthodox community, what they do is there's an organization called Dor Yeshurim. I don't know, did anybody hear about Dor Yeshurim before? So what they do is, you wouldn't think, because the ultra-Orthodox community, they tend to arrange marriage, and they are a close community. They are trying to avoid those genetic diseases that are typical for Jewish Ashkenazi community. And so, because it's arranged, they know who is going to marry who. What they do is that they collect DNA samples from, actually they go into teenagers just before they are being before they're presented to the community for marriage, and they everybody is giving a blood sample, and it's anonymous, and then they do everybody gets a number. They don't give, and they don't never get the results back, but they test individuals for the 38 different diseases that they have, and when there's a proposal for marriage, they compare the numbers and see if there is a compatibility between the bride and the groom. If there's a high chance of genetic diseases, they don't approve, or they don't approve the marriage. And if there's low chance of if there's a high compatibility, the low chance of they're not recessive for the same genes, they don't carry the same mutations, then they approve the marriage. This is all done anonymously through organization. It is actually was founded here in the United States, and I think they set it in Texas. It's set first for the ultra-Orthodox community in New York, because whoever set that organization, he had several kids that were suffered from Thai-Sachs, and he wanted to prevent it from happening to other families. And it's now all the ultra-Orthodox communities in New York and in Israel are using those services, and they approach the kids when they're still in high school before they are set up for marriage.
So what percentage of the people are free of, those are a lot of diseases. And if you're talking about a small community, it seems like they would be at high risk to have something, so.
Right. So I'm not sure what's the percentage in each different subgroup of the ultra-Orthodox, because they have all kinds of branches in there, and I don't know what's their, it's a close community, so I'm not sure how exactly they're into marriage, and is it like only, you know, Satmar is marrying Satmar, or, so I don't know exactly, but I know that they do use it, and then they do use the services.
And I don't know, correct me if I'm wrong, but of those 38, not all of them have as high a percentage in the
Right.
population
And it's only
as
2%.
Tay-Sachs. Some of them are very rare.
Yeah.
Right here, I think I'm showing some of the percentages.
And that's-
So, you know, for this, okay, this is for the Mizrahi and Sephardic Jews, but you can see alpha thalassemia, for example, is, these are high frequency, this is 1 to 5 and 1 to 100 in Moroccan Jews. But Tay-Sachs in the Sephardic population is only 1%. So not everybody has the same, and some of them are, as Mark said, are very, very rare. The other way to study population is to go through lineage, and luckily if the Y chromosome is transmitted only from male to male to male, because females don't have Y chromosomes, and so you can track down genetic information in the population through the Y chromosome, because you can always find relationship within the family between father to son who are having the same Y chromosome. So, for example, a lost tribe in Africa, as the Lemba tribe, they had, they live in Zimbabwe. There are about 50,000 people in that tribe, and they look like their neighbors, but they are claim that they are part of the Jewish people, and they kept some of the traditions of Jewish people. They kept the Sabbath. They kept. They had different ways to cut the meat, you know, and prepare the meat and so forth. And then when they were genetically analyzed, they used the Y chromosome and see how similar it is. And they found that in some of those priesthood, their Y chromosome looks very much like the Kohanim Y chromosome that is in other Jewish populations. So it looks like they were somehow maybe moved through from Yemen back down to South Africa, but the priesthood, the line of priesthood, stayed the same, and they share the same Y chromosome as everybody else who has a Kohen, who's part of the Jewish population. So that's one thing. And then another thing to measure relatedness in communities are looking at what we call mitochondrial DNA. So this is a DNA that is in our cells, but it's not part of the DNA that is in the nucleus of the cells, but it's in another organelle that is called a mitochondria. And the mitochondria is a small organelle in the cells that is responsible for respiratory production. And the mitochondrial DNA is only transferred from mothers to the offspring. So you can ascertain relationship because all the kids will have the same mitochondrial DNA as their mother, and the mother will have the same mitochondrial DNA as her mother. So you can track down relationship within families based on mitochondrial DNA through the mother lineage, through the mother side, and through the Y chromosome on the parental side, paternal side. So just moving into cancer. So cancer, in contrast to the different diseases that I mentioned before, is not just a single gene disease, but it's actually it's what we call a complex genetic disease, because there are many genes that are affecting cancer onset and the onset of the disease, and not just genes, not that just many different genes, but also impact from the environment, our exposure to environmental toxin, our exposure to DNA damage or to hormones and so forth. So all these together come to play in the initiation of cancer. And cancer, as I said, could be either genetically, there is a specific genetic component to it, but it's also an environmental component to it. And majority of cancers have some kind of a combination between the gene and the genetics and the environment. But there is some cancer types that if you have the gene, no matter what you have, how you try to prevent it, you are destined to have the cancer. This is, for example, a specific type of colon cancer. If you have a gene in the APC gene, you are for 100% sure will have a colon cancer. If you were exposed to Chernobyl and lived next to Chernobyl when they exploded, you probably have 100% of contracting cancer. But the rest of the cancer types, there's some kind of a combination between the genes and the environment. So breast cancer is the second common cancer for women, following after skin cancer, and it's affecting 1 out of 8 women. In 2015 alone, there were about 231 women who were diagnosed with invasive cancer, in addition to about 60,000 women that were diagnosed with local type of cancer, that is when it's not spread yet. And then it's expected that every year there are about 40,000 women who die from breast cancer, which is unfortunate. So what causes cancer? Breast cancer, as I said, there is a combination of both the genetics and the environment, and 90% of the cancers, the breast cancers, are due to somatic mutations, so mutations that occur because we're exposed to environmental toxins, we're exposed to hormones, and so forth. And about 10% are because of genes that are contributing in the germline. So these are genes that are inherited and contributing to breast cancer. Those genes are, I'm sure you heard about, BRCA1 and BRCA2. And then we have some other genes that are also less known and less and contributing less strongly to those for cancer. So BRCA1 was the first cancer gene that was identified as a hereditary cancer gene. BRCA1 stands for breast cancer gene 1, and BRCA2 was found a year and a half after BRCA1, and it's called BRCA2 because it was found second. And they both together contribute to between almost 10% of the hereditary breast cancer. So cancer that is run in families, they're almost 10%. But then we also have the other genes, P53, ATM, CHEK2, and so less. Yes.
Is the 50% number means that 50% of women with the gene get cancer?
No, this means that of the 10% of breast cancer that is because of inherited breast cancer in families that have breast cancer in the families, 50% about of them have a mutation in BRCA1.
So can I ask a related question?
Yeah.
Has someone been tested for the BRCA1 and the BRCA2? If you don't, can you go back? If you don't have those, you still have a 90% chance of getting breast cancer. Is that what that says?
No, no. I'll talk about the percentage in just a second. But so the general population is 1 in 8, so it's 12%. For every woman who, just being a woman, you have 12% chance of having breast cancer in your lifetime. If you have BRCA1, you have up to 80% chance of having breast cancer. Yes?
When you address your question, are you going to talk about what it means to have a negative test?
Yeah, I think so, yes.
So if you have a negative test.
If you have a negative test, that means, so, like, I think I do
You're gonna
address
get
it,
there, okay.
yes. Okay, so some of the risk factors, as I said, are genetics, and and you cannot modify them. Your family history, your genetic risk, your personal history, or your menstrual history, those are things that you cannot modify. They are just there, and they contribute to risk or not. But then there are also modifiable risk, is just environment, reproductive history. If you have, the more children you have, and then you have less chance of breast cancer. If you have hormone replacement therapy, can induce some, can promote some cancer. Diet, alcohol, all these are modifiable factors. So the BRCA1 gene was identified in 1994, I believe, and it's found on chromosome 17. And this is a schematic presentation of the gene. It's a large gene, and you can see all these red and yellow cubes are representing different mutations. And in fact, there were more than 500 different mutations that were reported for BRCA1, and they have reported many, many, many times. There's a database that is held by NIH that collect all the data about different mutations in different populations and so forth. The protein itself, the BRCA1 gene, is code for a protein that is called BRCA1, and the protein is actually involved in trying to repair DNA and make sure that the DNA has no changes and is intact, so it helps maintain the integrity of the DNA. The BRCA2 gene was identified about a year and a half after the BRCA1 gene, and it's on a different chromosome, and it has also multiple mutations. It's also a very large gene, and it also functions in the DNA repair pathway, so it also helps to maintain genomic stability and integrity of the genome. Even though they are they're both working in the same pathway of DNA repair, and there's both large genes, there's no relation in their structure. The two genes don't look alike. They have different structure of genomic data in there. So right after the gene was for BRCA1 was identified, there was a study that was actually done here as a population study that was done here in Washington. I don't know, in mid-90s. They were collecting DNA from Ashkenazi women in different communities and synagogues in the Washington area. And based on that, they were able to ascertain that if you have a genetic mutation in BRCA1 and BRCA2, you have a lifetime risk of up to 70 or 80% of chance of being diagnosed with breast cancer. This was later on also validated by other studies, and as I said, non-carriers they're not free of risk. They have about 12%. They have 12% risk of being diagnosed with breast cancer just because they are women. So by age of 70, if you have BRCA1, you have up to 65-70% chance of if you have BRCA1 mutation, a little bit less chance if you have reduced risk if you have BRCA2 mutation, and then as I said, if you have normal BRCA1, you have 12% of. Yes.
Can I ask a question just to make sure I understand you properly? So am I correct that everyone has these genes, it's just whether you have mutations on
Right.
the genes?
So everybody has the gene because we all have the same 20,000 genes in our genome, in our body. But some of us have mutations in particular gene, and mutations in BRCA1 increase the risk of breast cancer by up to 70%.
Okay, and then just very quick follow-up. You said there are hundreds of different types of mutations that have been found.
Right.
Do they all carry the same increased risk? Or do you
No,
know
so
that?
there are different mutations have different risk ascertained to them, and I'll just talk about them just in a second.
And just to be clear, each affected person only has a single mutation.
Right.
So the 500 are different ones in the entire
in their population, right. So every person who has BRCA1 mutation has one mutation, and because it's a dominant condition, you have only to have mutation only in, you're at risk only if you have, you're at risk even if you have just one copy mutated, not the 2 copies mutated. This is basically showing the same, that if you have BRCA1 mutation, you have an increased risk of up to 70% during your lifetime. And if you have BRCA2 mutation, you have, and actually, if you, and also increase the risk of ovarian cancer to up to 60%. But even though it's hereditary, yes?
Since the BRCA1 and BRCA2 are different genes, can you have a mutation in both?
So there are not a lot of information, there are not a lot of cases that would have mutations in both, but I know of some, very few, that have one mutation in one gene and a mutation in another gene, but it's not very common. So even though it's a genetic disease, there's still some impact of the environment. And it was found that if you were born before 1940, you actually have a lower, with the same mutation, you have a lower risk of being diagnosed with breast cancer than if you were born after 1940. And it's probably some kind of a changes in lifestyle and environment. And then if you had, if you were never pregnant, you have a higher risk of being diagnosed with breast cancer with BRCA1 mutation than if you had been pregnant at least once. And then also if you were leaner when you were adolescents, then you have a lower risk of having breast cancer with BRCA1 mutation than if you were heavier and less active. So there are other contributing factors for being diagnosed with breast cancer that modify the risk. So even if you have the same mutation, different women with the same mutation may have different risk because of the different, because exposure to different environmental factors and changes in lifestyle and so forth. So in the Ashkenazi Jewish population, there are 3 mutations that, 2 in BRCA1 and 1 in BRCA2 that contribute to the high risk of breast cancer and ovarian cancer. And altogether, if you add the carrier frequency, they add to 2.4 or 2.5, so means 1 out of 40 women have a mutation either in BRCA1 or BRCA2. Those 3 mutations account for 90% of the mutations identified in the Jewish Ashkenazi population, which means that if you were not diagnosed with one of these mutations, you're still at risk for being having a different mutations in BRCA1 or BRCA2. A lot of times in the Jewish Ashkenazi population, they because there's a high frequency of those mutations, they're tested only for those mutations. They're not tested for other mutations. And then if they don't find those mutations and you still have a strong family history, they're being the whole gene is being sequenced to find if there are other rare variants in the genes. And sometimes they could be rare variants, and sometimes they could be some structural changes in the chromosome that contribute to for breast cancer risk. But those are.
Sorry, because this is what I meant. What it means for a negative test. So it depends on who did the testing, when you had the testing done. So not all mutations are bad. I do want to make that clear. We have 6 million differences between you and me. Not all of them are causative for disease. The ones that we're testing for today are ones that we know based on databases of individuals who have had testing done in the past. Some companies have made that available. Others have not. And so there are efforts underway in the public domain to try and collect a lot of this data. So it depends on who did your testing and when it was done. And even what I mean by when, because there have been different offerings over different time periods. And so as she was saying, because you had a negative test, it means they didn't score any of the currently known mutations at the time of your test.
So the more recently you had it done, the more likely it is that it was accurate?
No, accuracy is a different.
I can keep worrying, okay. That's what
Accuracy is a different question, and I won't get into what that
Okay,
means
okay.
here.
All right.
But it just means that, it depends on what was screened.
Okay.
Okay.
Okay, thanks.
So in Israel, if you're Ashkenazi Jewish in Israel, you're only being tested for those three mutations. And testing is actually covered again by insurance because we have a national health insurance. So it's if you have indication of family history, you're being tested for those. And in the U.S., I'm not sure if I think if you're Ashkenazi Jewish, you're also being tested first for these, and then if they come negative, the genome is being sequenced and to identify rare variants. I think today they're moving into health insurance companies here also. If you have a strong family history, and I think I mentioned it in one of the next slides, the health insurance will cover it. And some of the companies actually will cover also the deductibles for the patients to help with the screening. So for the Jewish Ashkenazi population, there's about 1 to 40 frequency of women. 1 out of 40 have mutation in BRCA1, is a carrier for BRCA1 or BRCA2. And just to make sure, because I'm saying that as 70% lifetime risk, that means that 30% that if you have a mutation, you might not ever be diagnosed with breast cancer, even if you have the mutation. So there's still variation within the population, even if you have the mutations or not. And the general population in the U.S. is 1 to 400 that are carrying a mutation in BRCA1 or BRCA2 gene. But those mutations, because of the nature of the American society and how it's very diverse, the mutations, they're not some strong hotspots like those 3 mutations for the Jewish population, and the screening or the identification of mutation is a lot more elaborate because you have to sequence the whole gene, and as I said, the gene is very large, so it could be very costly.
Ronit,
Yeah.
in the non-Ashkenazi population, are those specific mutations ever found?
Okay, so that's a good question. So there are mutations that are typical for Sephardic Jews or Iranian Jews and Afghanistan Jews. There were identified founder mutations on those population, and in the Iraqi Jews, the mutation, the 185 LINEAGE that is common in Ashkenazi Jews is also common in Iraqi Jews, which probably means if you look at like a history and you can map it so that maybe that mutation arose before they split into and moved into different continents when they were expelled from Israel. And this is just to show you that other populations also have mutations in BRCA1 and BRCA2 that are frequent. We have typical mutations that are what we call a founder mutation in the Icelandic population. They have 1 in BRCA2 and 1 in BRCA1 that is a little bit less common. And French Canadians have at least 6 different mutations in BRCA1 and BRCA2 that are considered to be highly prevalent, and then there's mutations in U.S. and Hispanic community in the U.S. and mutations that are in Black Americans. And the Black Americans, initially, they didn't find any mutations. They didn't think that they are BRCA1, BRCA2 carriers, even for aggressive breast cancer. But now they're finding that they sampled the population a little bit differently, and now they're finding that women with African Americans also carry mutations in BRCA1 and BRCA2.
Can you comment please on how the sampling differed? Because that's a pretty dramatic difference.
Right. I think what they did before is they used criteria of really strong family history, and they didn't find mutations, and when they made the criteria less stringent, they found more mutations, because those mutations maybe weren't so penetrant or so strongly increasing risk, but they were there. And yeah. So turn out ovarian cancer is high impacted by mutations in BRCA1 and BRCA2, and 39% of women who are diagnosed with with ovarian cancer actually have mutations in BRCA1. 17% of ovarian cancer patients who have mutations in BRCA2, and if no mutations, just in the general population, the risk of women being diagnosed with ovarian cancer is 1.3%. So women diagnosed with ovarian cancer are more likely to have actually being diagnosed with, have a mutation in BRCA1 or BRCA2. So the frequency of mutations in ovarian cancer is higher relative to the contribution of those mutations to breast cancer. And just to add on, not just BRCA1 and BRCA2, there's other mutations, genetic mutations that contribute to ovarian cancer, and there's a syndrome called Lynch syndrome, and there are several genes that are contributing to, that are mutated in Lynch syndrome, and those genes also participate in DNA repair and maintaining genomic stability. And those mutations that contribute to colon cancer and ovarian cancer, and they are also frequent in the Jewish population. So what are the criteria for testing for BRCA1 and BRCA2? So if there's a breast cancer that was diagnosed before age of 40, that is an indicator for testing. If women had cancer in both breasts, if there are both breast and ovarian cancer within the family, if there are multiple recurrence of breast cancer, that's another indication for testing. If there and then if there are cases of male breast cancer, and if you're Ashkenazi Jewish, it's also indication for testing for BRCA1 and BRCA2. And then, as I said, because the frequency of mutations in ovarian cancer is so high, ovarian cancer is also an indication of testing for BRCA1 and BRCA2 in some cases. So these are criteria for testing, but what's coming out now, because the frequency of the BRCA1, BRCA2 in the population of Ashkenazi Jews is so frequent, there are now suggestions, and especially it started in Israel, that maybe instead of just testing, maybe the whole population needs to be screened up front, even before you have symptoms, just to make sure that that you can try and prevent the occurrence of breast cancer. So screening, we're all screened for breast cancer after 45. We all go do our mammograms. So it's not new at the concept of screening, but the question is of genetic screening. And so in Israel, they found out, they actually did, they collected male Ashkenazi males they recruited Ashkenazi males, and they were able to recruit 8,100, about 8,200 males that were Ashkenazi, and they sequenced their BRCA1 genes. And these are not necessarily males that had breast cancer, but they just presented because they recruited for a study. And so it turned out that 175 of those 8,200 males had BRCA1 mutations, even though they didn't have any family history in particular. And then 50% of them didn't have family history. And then they looked at those males that have BRCA1, BRCA2 mutations and sequenced and their female members of their families to see if those mutations are in the family or just happened in the males. And they found that 211 female mutations carriers that were identified that are related to those male that were recruited initially. So based on that, they figure out that they are actually able to identify mutation carriers that were asymptomatic in the population and no strong family history. And it's really tricky with Ashkenazi Jews to have a strong family history because of a lot of our families are small and some of them didn't survive the Holocaust, and we don't have a way to track down what happened before. So not having a strong family history may not indicate that you don't have it in your family. So based on this study, now they're recommending, yes, they're now recommending to actually genetically screen all Ashkenazi Jews in Israel. There were studies that performed later on in the UK after this, and also found out that they were able to identify mutation carriers that had not been identified otherwise because they didn't have strong family history. Yes?
So I'm curious about the 211 mutation carriers that were identified. Did they test descendants and also like parental units? So for the men who tested positive, did they test like if the mother was alive, did they test the mother or the father and also if they had children?
Right, so these 211, they came out from family members of the males that were tested positive.
Older, younger siblings?
Right.
Okay.
Yes.
And you've used the word testing and screening in the last 3 minutes, and I'm not sure which is which.
So testing is, before, testing is like if you had all those indication, if you had a strong family, if you had multiple recurrence of breast cancer, you would be sent for testing to see if you're a BRCA1 or BRCA2 carrier. What they're doing here is that they are screening, so they're doing a genetic test, but up front, before you have the symptoms. So
it's the same procedure.
it's the same procedure, but now they're looking for
Different population.
it's a different population. So these are female that haven't had breast cancer incidents yet, but they were diagnosed based on those genetic tests. I guess testing a genetic test is that they were diagnosed that they carry mutations, but they were asymptomatic at the time of the test.
It's a little confusing, it's the same exact thing.
Yeah.
This
This is just population screening. So for instance, when anyone who here who had kids, if you remember, they did newborn screening, did a little heel prick, took some blood, and ran tests. That's screening. Those are kids that we don't know have a disease necessarily. So we're trying to catch diseases that we can't detect. We want to be able to catch that early in case we can treat it early. So that's a population-based screening. It's still the genetic test. That's not a genetic test, but what she's talking about is still doing the test, but it's in a screening basis.
You're
Not-
still genetically
not
doing the
people-
test
not
of
for
the
people who have a disease. So-
but you're screening the population based on just because they are Jewish and Ashkenazi, not because they are showing any symptoms or you suspect that they are breast cancer patients. Yes.
Can you talk about the relationship between screening and mortality? Like why screening?
Okay, I think it's coming up. Okay, so what's the implication of finding that you have BRCA1 or BRCA2 mutations? So if you're a carrier of BRCA1 or BRCA2 mutation, you are at high risk of developing multiple recurrence incidents in ipsilateral, means in the same breast, or contralateral in the outer breast, developing other incidents of breast cancer or ovarian cancer. BRCA1 tumors tend to be triple negative, which means that they don't have several of the characteristics of the general population breast cancer. so they can not be treated with the currently used drugs for regular breast cancer. If triple negative means that they don't have estrogen receptor, they don't have progesterone receptor, and they don't have the protein called HER2. Current treatments for breast cancer in the general population is to treat with tamoxifen or other estrogen receptor modulators to stop the estrogen receptor from working, and if you have a HER2 protein, you get a Herceptin. If you don't have any of those, you cannot be treated with those three. If you don't express the estrogen receptor, progesterone receptor, or the HER2 protein, there's no point of giving those medicine for you. So you have to have a different course of treatment. And until recently, there was not much of a treatment options for triple negative breast cancer. You had to go directly to chemotherapy. Now they're developing modalities that will target triple negative breast cancer, and some of them are working, and then recently there's a drug that was approved for ovarian cancer of BRCA1, BRCA2 mutations that is called PARP inhibitor, and so they're being still tested in clinical trials for breast cancer. If you have BRCA1, BRCA2 mutation, your other family members are at increased risk for, males are increased risk for prostate cancer, and there's an increased risk for pancreatic cancer. So it's good to know if you are a carrier, because you can then also you can monitor other members of the family. So I think I want to just go here, for example. Some of the options for reducing the risk will be to do surgery and do risk reduction surgery, and it could be either bilateral salpingo oophorectomy, so you're moving the fallopian tubes and the ovaries, and that reduces 80% reduction of ovarian cancer and reduction of breast cancer. Of course, if you're planning a family, you want to wait until you had your kids to do this kind of surgery, because it will affect your ability. And then if you're do not want to do such a radical, you can do a mastectomy, a double mastectomy, and that reduces the risk of breast cancer as well. It turns out that they found out that if you do those surgeries, and especially the bilateral splendorectomy at a young age, it basically induce postmenopausal symptoms. So if you're young and you're showing symptoms of you have postmenopausal symptoms, you can be treated still with hormone replacement therapy, and it doesn't negate the effects of or reduce the effect of the bilateral oophorectomy. Then if you know that you're a carrier, you can also monitor yourself by doing screening, doing MRIs, or you can use what they do now is doing treatments for prevention, or what we call chemoprevention, and providing, for example, tamoxifen to women who know that they are carriers. Somehow, even though they don't have estrogen receptor, it was shown that it works as a preventive measure. And so this is another option. So there are benefits to know. You can do something about it if you know that you're BRCA1 or BRCA2 positive. Going backwards, I just want to. Okay. So because as I mentioned, that BRCA1 and BRCA2 are the proteins themselves are important for maintaining intact DNA and are important for DNA repair. If you have drugs that actually cause DNA damage, a lot of the chemotherapeutic drugs are actually causing DNA damage. So actually BRCA1, BRCA2 respond better to chemotherapy than other patients of breast cancer, and those tumors are more sensitive to chemotherapeutic drugs, especially cisplatin. And as I mentioned, PARP inhibitor is a new It's a new drug that is now available for ovarian cancer and still clinical trials for breast cancer, and it basically inhibits DNA repair. So it basically enhances the sensitivity of tumor cells to go through, to die, because you're preventing the cells from any options to repair their DNA, and then eventually they can't repair the DNA. There are so many mutations that tumors have to die. So is breast cancer a Jewish disease? I hope I convinced you it's not. It is more frequent in Ashkenazi Jewish, but it's really the relative risk that it's being added is not significant. It's not very significant, and if you do the math, and there are maybe 5% more or less of mutation of breast cancer incidents that are due to BRCA1 in the Ashkenazi population, other than what it would be in the general population. So some of the benefits of doing not just BRCA1, BRCA2 mutation, but screening for all kinds of variants in the genome is that we can find new drugs and new treatments. We can personalize the treatment and do specific treatments for specific tumors. They may help to diagnose and optimize the treatment, and also identify diagnostic markers and prognostic markers, and help also with prevention. So there's a lot of benefit of identifying what is in your genome, and for this kind of cases, and just in general. So I think with that I will finish, and if you have any more questions, I'll be happy to answer.
Can I ask a quick question?
Sure.
So you said those are the benefits. Can you just mention some of the risks, either to the individual or the family? And I know, I mean, there's an obvious answer here of, you know, you have a gene and you're sort of asking your whole family to be tested because you want to know the risk in the family, or you want to make people aware not everybody wants to know. And how do we as a society make decisions about who gets to know my genetic information or your genetic information? How much do you impose on your relatives, and how much strife that causes? I mean, these are the ethical issues which I deal with that have nothing to do with science, but have to do with how do we talk about these risks, and how much can you put your foot on somebody else to take the test when that person doesn't want to know every piece of information, especially when there's not that much you can do for certain kinds of diseases at this time.
Right. Yeah, it's a very important question. So because if you find out, do you tell the rest of the family, or do you not? What if they don't want to know? It's exactly the questions that you raised. What's your obligation to tell them? I don't know that there are real answers for that. It's right or wrong. I think if somebody doesn't want to know, I think you have to respect it. But then, what do you? Yeah.
But there are also scientific, actual risks associated with surgery or drug therapies for people who are asymptomatic. So that's an addition. So you have a choice. If I know I can have a double mastectomy, I can have a preventive tamoxifen, I can do those things. But those also carry risks. So there's a
Right. So there's always the active balance, right? It's like, what's the risk that was having not doing anything, or what's the risk of doing proactive? But you can also just monitoring it and doing annual MRIs. That doesn't carry a lot of risk, but at least you can monitor it and better, and then take action sooner, like find out earlier, and that's always helped to diagnose earlier than later. Yes.
Cost and how you get different screenings? I mean, can you walk in off the street? Could you go into that?
So I don't think so. I think you have to have, as I said, some of the risk factors. So either it's in your family or you had multiple incidents of cancer already diagnosed. And so I don't think you can just go into your physician and ask, okay, I just want to be diagnosed, tested for BRCA1 or BRCA2. I think in Israel it's a little bit different because if you're Ashkenazi, I think it's now they're offering it to everybody to be tested. Here, I think the insurance, I know that with the Affordable Care Act now, it was provided if you, it was covered. I don't know what will happen next.
So I can respond out of my own experience. So my GYN and I have been discussing it for years because of the very high incidence in my father's side of the family, which they didn't used to think was a high marker. But then when my second cousin was discovered with breast cancer, and she and I are the next, there were no women in the intervening generation. I got a copy of her test and she was BRCA positive, and then my GYN said, yes, you should go talk to a genetic counselor. But probably only if you should probably only, and then she and I talked at great length, and of course I'm very nervous, but you should probably only take the test if you're willing to do something about it. That's the scary thing about information, but then there are all these other things you can do, like just more frequent monitoring. And I was lucky enough to come back negative, but I'm still sitting, and it was $500, and I'm still fighting with my insurance for covering it. Okay, even though my doctor said that was the other thing. So I'm still encouraging my siblings because of the, she said before she did the test that based on my family history, I had a 25% chance of being positive, which sounds pretty high to me. Yeah. And, but I'm still urging my siblings because they each have 2 daughters to follow up.
So can I add one thing?
Sure.
So there are some protections by law for genetic testing. This was codified in the Genetic Information Non-Discrimination Act, but it does not cover all situations. So for instance, life insurance and long-term care, if you have a genetic test, you are not protected in those situations. So it's just something to be aware of if you're considering or talking to your siblings about it, that they're not going to be, there are certain places where they will not receive protections under law right now.
Privacy of the information being given, is
That it can be used in determining whether you can receive that by a company. So for currently, yeah, so long-term care and life insurance are not covered.
So
I
it's
will say
health
that
insurance that's covered.
Health insurance is covered.
And employment are covered, but long-term care and life insurance are not.
Health
Certain
insurance
states
covered.
have done stuff on a state-by-state level. You need to check on your state to find out what protections have been put in place in that state.
Very important.
So just in case anyone's thinking about running out, I just want to make sure you're aware.
So can I just re-emphasize one point that Ronit made early? There is no such thing as a Jewish gene.
Yes.
We all have the same genes. We all have a gene for hemoglobin, for instance. But there are individual mutations that are more frequent in the Jewish population. There are, in the case of hemoglobin, there are mutations that are more frequent in Sardinian populations or in African populations because certain mutations protect against malaria. So it's the individual mutations that are associated with population, not the gene. There's no such thing as a Jewish gene.
Not the religion, just the population. Right. Not the religion, because you're in the population.
Right, and I think Jewish is kind of unique because Jewish, you don't have to be religious to be Jewish, part of the Jewish religion, right?
Yeah, you're preaching to the choir, you don't have to be Jewish.
Right, you still.
So let's take one more question.
Yes.
So I have a question about some of the requests that are out there for further testing. There's one that's going around now, for example, for kidney disease, looking for Ashkenazi Jews, and I also had BRCA testing, also fortunately negative, and mine was paid for because my grandparents were first cousins. There was a male in the family with breast cancer, pancreatic and GI cancer, so that was different than.
No, I think they might come around. They
differences that were identified that were not of clinical significance, but they said if you'd like to participate in a study. Can you give a little information about this information that we all occasionally see and what the usefulness might be of us participating in such studies as part of the Ashkenazi population?
I'm not sure because, what would you do with the information? I'm not
Well,
sure.
it's more just to contribute the information to the research.
Right, and so it does run to a risk like, sorry, what you said is that, your health insurance is covered, but your life insurance and, your.
So when you are participating in those kinds of studies, are you identified or are you not?
Oh, so,
And
so
the
these are like
information
research
is-
studies?
Yeah.
Oh,
And the
okay.
information is not necessarily
No.
returned to you.
That's right.
It will,
Right, so-
it will
right.
say in
Okay.
the,
Uh-huh.
in the,
Informed consent.
Oh, okay.
informed
Okay,
consent,
so-
whether or not. So
But
that,
that's not a risk added to our insurance, right?
No.
no.
No, but then you might not find out of the information.
Yeah, you're not gonna find it.
Yeah.
So presumably the
Exactly.
primary motivation for participating in a study like that is altruism.
Right. It's just to contribute to the common knowledge.
Well, thank you very much.
Thank you.